Okay, so Shubham Chhajed, hi, thank you very much for agreeing to talk to us tonight. Shubham is the recipient of the 2020 Val Williams Scholarship, and the title of his thesis is Hydraulic Drivers of Photosynthetic Variation in Co-occurring Plant Species in the Sydney region. A least cost theory approach.
So, like Sue mentioned, my focus is on looking at how plants manage their water budgets. Basically, different species of plants, they all use the same resources for photosynthesis. They all use carbon dioxide, water, nutrients, and sunlight. And still, there's so much variation in how much plants photosynthesize, and a lot of this variation is not explained. And I'm trying to understand if how plants manage their water budget, or the different strategies that plants have in order to use water. Does that have anything to do with photosynthesis and just carbon fixation? So today I'm going to talk about saving water or spending it, and how plants use water. So just to begin this talk, I wanted to ask a question to all of you. What is the costliest thing that you bought last year? Just if you want to unmute yourself or put things in the chat, it's fine. Just a few ideas. Awesome. I'm seeing some options in the chat: a house, food, windows, clothing, perhaps tissue roll. Anyone? So we all know that tissue paper is really cheap in normal times. But last year in the COVID crisis, we saw that the prices went really high. And I'm sure everyone saw videos of people just fighting each other over a roll of tissue paper. And so when a crisis hits, things which are seemingly very cheap become extremely costly, just because there's not enough of it available in the market. And so the term "what's costly" is in fact very relative to what conditions we are talking about. And there's a similar bargain or similar exchange that happens in plants. So if we look at a leaf, there are these small, tiny windows, which are called stomata, which allow the leaves to take in carbon dioxide. But in this process, they lose a lot of water, just simply because the tissue inside is very humid, and the dry outside is very, very dry. The air outside is very dry. And just because of that difference in concentration of these molecules, whenever these windows or these stomata open up to take in carbon dioxide, plants inevitably lose a lot of water. And on a normal day, this exchange could mean for every gram of carbon dioxide that a plant is taking in, it can lose up to 300 grams of water. And when things become really harsh, when it's very hot or very dry, this 300 number can go easily up. For example, let's say 400, but it's not restricted to 400, and it can easily shoot up. So whenever there's some sort of gas exchange, there's an inherent trade-off or a bargain of carbon versus water. And this becomes very important in conditions like drought. So we all know Australia went through one of the harshest droughts it has ever seen in the past 200 decades. This was the harshest drought that Australia saw. And this is a photo from a eucalypt forest in Armidale. And we can see that all these trees are showing a lot of browning of leaves, which is just the huge amount of canopy death that we saw. And whenever such conditions exist, there's these stomata close themselves so that they don't lose a lot of water. And but before that happens, whenever the stomata is open, there's a whole hydraulic system which is responsible for transporting water from the soil to the top of the canopy or to the leaves. And it is very essential that plants maintain this continuous supply of water so that it can open its leaves, carry out photosynthesis, and go about it's going to go on about its daily business. But whenever these conditions, there are these extreme environmental conditions like drought, especially like the one we saw in 2019, this water pathway comes under extreme pressure. And under this pressure, there's air bubbles that can get trapped in this pathway. And under higher amounts of these air bubbles, they can easily block this flow of water. And this event is called embolism. So if we just think about emboli in humans, which block blood flow in human body, it can lead to severe effects like heart attack or hemorrhages, or maybe some of them can be very fatal. And it's the same in plants as well. So under smaller amounts or smaller concentrations of embolism, there's a reduced flow of water, and it can hamper photosynthesis. So it can reduce the level of photosynthetic efficiency that plants display. Here in this example, I'm holding a small stem sample, and you can see some air bubbles here coming out of the water. So I'm putting some pressure on the other end of the stem. There's some water under pressure, and the water that comes out pushes the air along with it, and we can see these air bubbles coming out as well. But when these air bubbles become really concentrated, they can lead to complete failure of the hydraulic pathway, and it can also lead to canopy death, which we saw here in this scenario. So now, I don't know about you, but I think that plants are pretty magical in the sense they surely should have some magic tricks up their sleeve to combat this evil of embolism. And my study is looking at what different strategies can plants apply to sort of avoid these embolisms and carry on photosynthesis, and just so that we can see how what strategies plants can use to sort of immediately counter these effects or carry on photosynthesis in over longer time periods. And one such strategy that I am focusing on for my PhD is water storage. So water storage is a very important factor in plant water dynamics, which has been heavily ignored whenever hydraulic pathway is concerned. In fact, up to 50% of the daily water needs that plants have can be met sometimes by water that is stored within the plants. So that means that not all the water that plants are transpiring or losing to the air or using during photosynthesis is coming from the soil all the time. And this stored water is often stored in tissues like parenchyma and fibers, which are surrounding these vessels or conduits or columns that carry water from the soil to the leaves, from the roots to the leaves. And so we know that this stored water can help mitigate some water needs. And so I wanted to check if the stored water reserves can help in avoiding embolism. And the way I do it is I measure the reliance of plants on stored water. And it's measured in kilograms of water that is stored per unit volume of sapwood, or the functional part of the stem, for every unit change in water potential. This water potential is basically a representative of the water demand that plants exhibit. And this is what I actually mean by reliance on stored water. So as on x-axis here, as we see that water demand rises from, if we take this point pre-dawn to midday, which is basically telling us before plants see any sunlight during the day, what is the water demand at that point, and what's the water status of the leaf. And then as the day progresses and things become drier, we see there's a higher level of midday water potential, which is even more negative and more demanding in terms of water. So as we dry the samples, I dry them on a bench, and then I put them in this water bath, and then I measure what the humidity is, or whatever. I measure what the potential of water. And then I periodically measured how much water was being lost. And this lost water is coming from stored reserves within the stem. And so this curve is telling me how much water the stem is losing over as the water demand is rising. And the slope of this curve between these points is basically how I measure capacitance, which is the reliance on stored water. So it tells me if the slope is steeper for a species, it means that more water is released to supplement the water demand. And the water demand I calculate by the difference in pre-dawn and midday water potential. Pre-dawn is when things are the coolest during the day, or before the day, in fact. And midday is when the sun is right above the plants and it's very demanding for the plants in terms of water. And so my research questions were to see if this capacitance, or this reliance on stored water, if this could lead to lower levels of blockage by air or lower levels of embolism. And then I also wanted to check if this higher capacitance allowed for photosynthesis to be maintained at a higher rate in plants, which meant more carbon fixation, or if this capacitance or stored water within the plants can help plants to photosynthesize longer into the dry periods. So for example, if things got really hot at 10 a.m., some plants would shut their the stomatal windows, and then stop photosynthesizing. But if they had more water reserve stored in their stem, they could use that stored water to sort of continue photosynthesizing, continue fixing carbon till, say, 12 p.m. or till noon or 1 p.m. Later during the day. And this is where the Val Williams scholarship funds come in. This was this time component of calculating if capacitance has a long-term effect was really only possible because of the funds that I got from the scholarship, which I'm very grateful for. The type of measurements that are required to measure that long-term effect are done by measuring isotopes of carbon in the leaves. And it's a very expensive analysis. And I had to send my samples to Australian National University. And so all of the funds went into that measurement for all the different plants that I studied. So in the rest of the talk, I'm going to talk about some of the plants that and the site that I studied where I tried to answer these questions, and then what are some of the results that I found. And like Sue mentioned earlier in the introduction, I worked at Castlereagh Nature Reserve, which has Cumberland Plains vegetation in the Sydney Basin area. So if we look at Blue Mountains, if you're going from Sydney to Blue Mountains, this Castlereagh Nature Reserve site comes just before you hit the boundary of Blue Mountains. And it's a small remnant patch surrounded by housing area, houses around it. And so but it's been conserved and under the protection of Office of Environment, New South Wales. And so it's been really great site for testing these questions because it has high Australian flora diversity. And it has very unique features which are useful for my study. So for instance, it has moderate amount of rainfall, around 800 millimeters per year, and moderate amount of soil phosphorus levels, around 200 parts per million. And it's only 30 to 40 meters above sea level, and it's pretty flat. And so all these factors meant that I won't be seeing any confounding variables that might be responsible for the type of measurements or anything else that I'm seeing. So I could really focus on what the differences are across species in the types of questions I'm asking. And a little bit about this site. This is a photo of the site where you can see a lot of eucalypts here. And it's dominated by ironbark and scribbly gum. So this is the front part of the site, and down this track, it's more of scribbly gum or white smooth bark. And there's a lot of other species in here that I was able to sample. So some of the species, so in total, I sampled around 18 most abundant woody plant species. And some of them were hairpin banksia or Banksia spinulosa, finger hakea or Hakea dactyloides, red ironbark, which is Eucalyptus fibrosa. I also had some other eucalypts, Eucalyptus longifolia and Eucalyptus racemosa, which is a scribbly gum. I also had 4 acacia species, Acacia podalirifolia, parametensis, Acacia falcata. There was also she oak, Alocasharina, a few Corymbia, Dodonea, and Persunia, and some other species. So I'm sure some of you might be aware of this flora and might know more about them than me. I've just been introduced to these species over the last couple of years, and I'm really still blown away by the diversity that a small patch in the middle of the city can show in terms of Australian plants, native plants. So these are some of the photos of me measuring stuff in this field. This is again another photo of the site, and there's many other species which you can see here. There's some scribbly gums which were not visible in the first one. So there's some smooth barks here. And this is me measuring the water potential or the water status of the leaves in the before morning at pre-dawn at around 5 a.m. And then we measured the same things again at between 12 and 1 p.m. To get the other extreme of the water status of the leaves. And then we used a Lycor machine, which helped us measure what the carbon fixation rates were for these plants and what the photosynthesis level was. And these are my lab mates who helped me with some of my measurements. And then we brought back samples to the lab, some leaves and some branches. And then I measured the capacitance or the stored water reliance in these species in the lab. And then I also measured what the amount of embolism was, or the amount of air bubbles in these stems. And for that, and so that brings me to the first question of my study, which is, does higher capacitance lead to lower levels of air blockage? And this is just a quick photo photo of the setup that I used to measure air blockage. This setup is in Western Sydney University, and I could go there in our collaborators lab and measure some of these traits for these species. So here there's a flow meter, which is attached to a water source, a syringe, and there's a stem sample here. So the water from this syringe flows through the flow meter and then through the stem. So this flow meter calculates how fast the water is flowing through the stem. And then we put the stem, which is here, under some pressure. So there's water flowing under around 100 kilopascal. And what this does is basically it flushes out all the air because of that much pressure. And then we measure the flow rate again. So initially we get a measure of flow rate when there's air blocking the passage of water, and then when the air has been removed and what the maximum capacity of the flow is. And the difference tells us how much of the embolism there is or how much effect or blockage there is because of the air. And this is just a branch section that I've connected to a tubing, which is then connected to a pipe of water, which allows us to do these measurements. So as expected, we saw that plants which had high amounts of stored water, restricted embolism to very low levels here. So here there's higher amount of stored water, and then there's very low levels of embolism. But plants which had high levels of air blockage or embolism were the ones that had very little reliance on stored water. So they didn't use stored reserves that much. They didn't rely on them that much. So this was very promising. This tells us that some species of plants can use the stored reserves to combat this air blockage and sort of maintain a proper functioning hydraulic pathway in the stem. So my next question was to check if this meant that plants or species which had this higher amount of reliance on stored water could increase their photosynthesis rate. And we saw that was not the case, even though these plants which had higher stored water had lower levels of embolism. On average, all sorts of species the relationship between photosynthesis and stored water was not significant, which just meant that directly there was not any increase in photosynthesis. But then we also wanted to check what are the other effects of this stored water. So for example, it is very costly. It can be very costly to have this stored water in the plant because for this, you need a lot of storage tissues. So earlier when I mentioned parenchyma and fibers, plants need to invest in those tissues so that they can store a lot of water. So it's equivalent to maintaining a water tank in your house where you need to construct a tank and maintain it so that you can use water whenever there's a need for it. And to test if it was rather costly to have that storage or not, we looked at a theoretical framework called least cost theory, which basically looks at the stomata again. And we see that whenever these stomata open up in the leaf, it allows carbon dioxide, which is outside, to come inside the leaf. And this carbon dioxide concentration is measured so that we can know what's the difference between the carbon dioxide concentration within the leaf versus outside the leaf. Excuse me. So this ratio of CI, which is the internal carbon dioxide, to the CA, which is the atmospheric or ambient carbon dioxide, can tell us a couple of things. So for example, if there's a low CI-CA ratio value, it means that more carbon dioxide is being drawn in, right? Because the internal CO2 is going down, which means more and more carbon dioxide is being drawn in by the leaf. But this also means that the stomata is open at this time and more water is being lost because of that exchange. And so water is becomes rather costly. And so a lower CI/CA value will tell us if water is costly. And if there's a higher CI/CA value, which means these are very close to each other, these concentrations, it means that carbon dioxide is not being drawn in that much. And so plants, rather than invest in proteins to fix this, whatever carbon dioxide is already in. So they sort of improve the efficiency of the proteins, which are responsible for converting these carbon dioxide molecules into carbohydrate. So the plants have to sort of make a decision as to if they should invest more in water or if they should invest more in these proteins. And that is where the least cost theory comes in. So it says that plants need to balance these costs or investments so that they can optimize photosynthesis. And the best possible outcome is when the total investment is the least for a given amount of photosynthesis. So the combination where you can achieve a given level of photosynthesis for the minimum cost that you might have to pay. And so we wanted to check if this balance is chipped off by capacitors or the stored reserves of water. So in which direction does this go? The plants, if they have higher reliance on stored water, does that make water costly or does that make carbon costly? And so we looked at these CI/CA values, which we measured again using those Lycor machines that I showed a photo of. The same machine which measures photosynthesis gives us that value. And we found that plants which had here on this end have higher reliance on stored water were able to maintain a lower CI/CA value if you look at this blue line. But there were a few plants which had a weird relationship with CI/CA, or which told us a few other things. So these species up here are the ones which are showing very, very low fluctuation in the water pressures. So if we look at the water demand, the difference between the pre-dawn before sunrise and at the peak of the day, these species at the top had very less fluctuation. And so because there was very less fluctuation in the water demand, they didn't really need that much of stored water. And so they didn't really make water costly in that sense. They didn't really have to rely on it. But as these fluctuations kept increasing for some of the other species, we saw that the more they relied on the stored water, the costlier water was and lower was the CI/CA value. And then these were the plants which had the highest fluctuation in their water demands. And so even lower CI/CA values for them. So these fluctuations in water demand is when these stored water are basically useful. And so the more that these fluctuations, more stored water is used and more investment in these storage tissues have to be made. And basically that makes the water that's coming out of these stored reserves very costly. So we saw that on a short-term basis, if you look at instantaneous values of carbon fixation and water usage, these stored reserves end up being very costly, but do not benefit that much in terms of photosynthesis. And so we thought maybe it had a very long-term effect. So basically, maybe the plants did not really increase their photosynthesis levels, but what these stored reserves helped them to do was carry on photosynthesizing even when conditions became harsher, perhaps at the same rates, but just for longer time. And that's where I could fill in that gap in the analysis with the isotope measurements from the Val Williams Fund, which brings me to my last question for today. If higher capacitance allowed plants to photosynthesize longer into dry periods. So to look at that, let's assume a species has more stored reserves of water. And these reserves are mainly coming from the parenchyma and fiber and other storage tissues. And we already saw that these stored reserves help in delaying the embolism effect or the air bubble blockage. And this, if plants are able to delay this air blockage, they can photosynthesize longer into the dry period. Even when the conditions are harsh, they can sort of maintain their normal levels of photosynthesis. So if we consider it over time, plants are able to fix more carbon for the amount of water that they use. So we should be seeing more carbon gain per unit of water, but on a longer time scale. And this is what I measured using a delta 13C isotopic signature. It basically is measuring how much of different carbon are there in the leaf. So carbon comes in two forms. One is 12 carbon and 13 carbon. And based on this intake of CO2 and escape of water, over time, based on the physiological conditions of the leaf, the selection of 12 carbon versus 13 carbon is affected because of various different physiological reasons. And that can tell us if the difference between those 12 and 13 carbon can tell us which strategy plant has been using over time. And the effect of this stored water in delaying embolism was calculated by calculating something called a buffering capacity. And so this buffering capacity is basically a ratio of how much water you have in storage and how quickly that water is being used. So for example, if we have a 2-liter water bottle, and someone is using 1 liter of water every day, that bottle will last him for 2 days. But if someone is using only half a liter of liter of water every day, that will last, that 2-liter bottle will last for 4 days. And so, if you have 2 liters versus 4 liters of storage. So it basically measures how much water you have and how quickly you can use it. And it tells you a buffering capacity. So basically a high buffering capacity or a high tau can means that plants can delay these effects of environmental conditions, which can allow them to maintain more carbon dioxide gain per unit of water. So by that, what I mean is if the buffering capacity is high because of high reserves of water, plants should be able to gain more carbon dioxide for the amount of water that they lose. And this is what we actually find in our species. So we see that higher tau or higher buffering capacity leads to more carbon gained over time, which is this delta 13C value, the isotopic signature. And plants which had lower buffering capacity had less amount of carbon fixed over time per unit of water. So this brings me to the final slide where, so in conclusion, I focused on stored water reserves in plants. And we found that the stored water reserves have some very important role to play. The benefits of having higher amounts of these reserves is that it delays the effect of intense dry conditions by reducing the risk of embolism or the risk of air blockage. And it increases the photosynthetic gains or gain of carbon, which can eventually lead to more growth and better survival opportunity for the plants. But this happens over a long time. It's a long-term return. So the question was, why do plants not invest more in stored reserves? If there are benefits to having that stored water in plants, why do all species not do it? And that's because it is costly to have those stored reserves of water. Costly in terms of investment that the plants have to make in the storage tissues, like the parenchyma and fibrous tissues. And this increases the cost of water use on a short-term basis. So if initially, when we looked at the CI/CA values, we could say that if we look at the immediate effect of this capacitance, it rather ends up rendering the water very costly. And so on a short-term basis, it actually increases the cost of whatever water you're using. And this again is only useful when the water availability is fluctuating a lot. So in scenarios where there's heavy rainfall or there's water available in ample amounts all throughout the year, those species don't necessarily have to rely on stored water reserves. Rather, they might just spend the water that whatever they have access to then and there, because that's more beneficial. That will allow them to have a higher photosynthetic gain on an immediate basis. And so, if they don't have to rely on these reserves, it's probably better to just use the water when it's available. So even though these are the stored reserves have some costs and benefits to it, we saw that all of these species that I looked at Castlereagh Nature Reserve are actually very different in how much stored reserves of water they have. And then we have some data from other sites in Sydney. And I'm going to compare the results of my site with those and see if we see similar strategies being played out at different sites in different species of plants around Sydney. And like Sue mentioned earlier on, my goal was to do this study across Australia in Queensland and Tasmania. But rather than that, I had to focus on these species themselves that I've been studying because of COVID restrictions. But it would be really good the next step would be to compare the results of this site with other species from other places and other environmental conditions to see how well we can sort of find similar patterns there. With that, I'd like to thank you all for being a very patient audience. And I'd also like to thank my supervisors and my colleagues, without whose support this wouldn't have been possible. And then mainly to Australian Plant Society and for the Val Williams Scholarship, which made a major part of this research possible, which I'm very thankful for. Thank you. And I'm happy to take any questions you might have.